If there was a medical procedure that could save the life of one of your loved ones, but it brought into question your religious convictions, would you hold true to your convictions, or would you alter them in the hopes that the procedure would save your loved one's life? Stem cell research brings this very question into play. My name is Clayton Coonrod, and throughout this presentation I'm going to talk to you about some of the controversies surrounding stem cell research and why I believe it should be federally funded. So to understand why stem cell research is such a huge topic and why it's such a key question of controversy, you have to first understand the basics. So what are stem cell research? Stem cells are unique cells within the body with two unique properties. They're undefined, and they can divide indefinitely and differentiate into specialized cells within the body. There are two main types, adult and embryonic. The first type I'm going to talk to you about is adult. Adults are limited in their use because they're already coming from an existing individual, which means that they already have a specific purpose. So skin stem cells extracted from an adult body already has a function contained within the skin, so it only can be used in the skin of a new host. They remain inactive for a long time, so they're not as fresh and as young as embryonic stem cells. There are 5 main types: blood, epithelial, which has to do with the digestive system, the third being skin, 4th nerve, and 5 skeletal, which also can be used to replenish cartilage and fatty tissue within the body. The second type is the main point of controversy, which is the embryonic. This brings into question, when does life actually begin? They're found in the early developmental stage, such as within the umbilical cord and the blastocyte. The blastocyte is a small sac of cells, roughly between 75 and 200 cells. This sac of cells is the point of controversy for many religious groups, such as Catholics, who believe that human life actually begins at the blastocyte formation, Jews, who believe that as long as the embryo doesn't come from an actual living womb, it can be used for medical science, and third is Muslims, who doesn't believe the embryo is an actual person at all. Now, the main point of the Jewish people is that the fact that the embryos used for stem cell research actually comes from discarded embryos from in vitro fertilization. So, now let's talk about iPSC, or intrapluripotent stem cells. These are adult stem cells which have been reprogrammed back to the embryonic stage. These are crucial because even though that they have the properties of embryonic stem cells, you can't use them in the human body because they are reverted back to the embryonic state through viral infection, which means that if they were introduced into a host body, they would spread the viral infection, making the host sick. However, these cells have important qualities due to the fact that we can test drugs on them, and we can see how certain diseases would impact the human body. The benefits of stem cells are numerous, but specifically, you can make a specific regimen designed for a specific individual. So, you take the stem cells out, you know how a human body is going to react to whatever drug or something you're going to pump into it. And this is also beneficial when implanting tissue and organ donations from another individual. You can repair the damaged tissue with an individual, and you can bring new cures about. Certain diseases such as heart disease, stroke, diabetes, nerve damage, Parkinson's, leukemia, multiple sclerosis or MS, Alzheimer's, and AIDS. Even severe burns are treatable. Now we're going to talk about some clinical trials that prove this process. According to the Lancet, 24 patients with different stages of MS or multiple sclerosis, all with a prognosis of significant disease progression. Now, if you don't know what multiple sclerosis is, it's a degenerative disease that affects all aspects of the body. So you feel numbness, blindness, even muscle spasms. And so, what happened was they took stem cells out, they froze them, and then they blasted these patients with excessive amounts of chemotherapy. So after the radiation, they took the stem cells, thawed them out, and re-injected them back into the individual. And what they found was that 16 of those 24 patients, so roughly 67%, the progression of the disease had completely stopped. Now we're going to talk about clinical number 1, which this was about cardiac patients. So Woolston in Biotechnology in Context, 2nd edition, stated that cardiac patients that he had examined not only had improved cardiac function after stem cells were introduced in their body, but also new blood vessels had begun to emerge where the damage once had existed. Woolston also noted in Biotechnology in Context Second Edition that Italian researchers performed a similar study in which they extracted stem cells from the limbus, or the exterior of the cornea, and then inserted them into the cornea, and eyesight was restored to 76% of these individuals. So how was this performed? What they do is they take stem cells from a certain part of the body, and then they're cultivated in a laboratory under specialized conditions to prevent contamination. And then the stem cells are left to divide. And this takes a long time because you have to produce millions and millions and millions of cells for them to have an effect in the body. So what you do is you take those millions of cells and you inject them back into the part of the body that is damaged, where they need to become specialized. And from there, they're left to cultivate and repair the damaged tissue. This is crucial with organ donation. So one of the things that I talked about earlier was organ donation, and how this creates a specific regimen for a specific individual that can be used to cure a specific disease within that individual. This is also presents a lower risk of rejection between the organ and the new host. Let's talk about one of the negative aspects that came about due to President Bush limiting our use of stem cell research, and that was stem cell tourism, which is American citizens, since we weren't funding stem cells, they left the country and went to underdeveloped nations where there's not an FDA to impose regulations and such. So these individuals could be exposed to a whole list of diseases from the lack of proper medical care. Thankfully, in 2011, President Obama reasserted stem cell research. So, George W. Bush actually limited the stem cell research to 60 existing lines. However, only 21 of them were usable, according to the Center for American Progress, due to contamination and the fact that some of those lines weren't actually fully developed. Now, according to Biotechnology in Context Second Edition, President Obama pledged in 2011 $358 million to adult stem cell research and $126 million to embryonic stem cell research, indicating a change in the United States' place towards stem cell research. The states have always pledged funding. 9 states pledged funding. 60% of the funding comes from New York and California. And that's according to the Center for American Progress as well. Now, is U.S. support increasing? Yes, it is. In 2007, 65% of Americans, according to the Center for American Progress, supported stem cell research. That went up to 68% in 2014, according to health research funding. So stem cell research is the next wave of cutting-edge and medical innovation that can lead to revolutionary new cures for once thought untreatable or even a death sentence diseases. With the federal government fully supporting and funding future endeavors into stem cell research, the promise of a brighter and healthier tomorrow can be achieved. My name is Clayton Coonrod, and thanks for watching.